The second event of guest lecture series for medical students, which presented by Indonesian Physiological Society and supported by Federation of Asian and Oceanian Physiological Society and International Union of Physiological Sciences is about to begin. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning, Indonesia. Good evening, America. Honorable Speaker, Prof. Kim Barrett, Professor and College, members of Indonesian Physiological Society, and also all students from Muhammadiyah University of North Sumatra, Medan, University of Pajajaran, Bandung, University of Hang Tuah, Surabaya, and also University of Jember. It is a great honor to have all of you here. My name is Widya Wasityastuti from Gajah Mada University, Yogyakarta, and I am privileged to have this opportunity in addressing you all in this auspicious occasion. It is an outstanding opportunity for us to receive a lecture from Prof. Kim A. Barrett, a famous and well-known expert in physiology, especially in gastrointestinal system from California University, San Diego. We are truly lucky to listen directly to the writer of Ganong Physiology, one of mandatory references in physiology. And today, Prof. Barrett will talk about the new development in gastrointestinal physiology. To begin with, I would like to invite Dr. Ermita Ibrahim Ilyas, the President of Indonesian Physiological Society, to deliver the welcome remarks and introduce the speaker.
Okay, Bismillahirrahmanirrahim. Thank you, Dr. Lydia. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Good evening, Professor Kim, and good morning, all participants. First of all, I would like to give my gratitude to our distinguished guest lecturer this morning, Professor Kim Barrett, PhD, from University of California, San Diego, USA, for your willingness to spend your valuable time for us this morning to give a special lecture with topic New Developments of Gastrointestinal Physiology. Before we proceed to the lecture session, I would like to introduce Professor Kim Barrett. Professor Kim Barrett, PhD, is a distinguished professor in medicine from School of Health Sciences, University of California. Prof. Kim graduated from University College London for Bachelor of Science in Medicinal Chemistry and PhD in Biological Chemistry. Her honorable position including actually a lot, two of them, editor-in-chief of the journal Physiology from 2016 to 2021, President of American Physiological Society 2013-2014, has several awards from American Physiological Society, American Gastroenterological Association, and the Physiological Society. Professor Barrett has had many research activities and funding as a principal investigator, all regarding the gastrointestinal system. 3 latest publications in 2020 and 7 publications in 2020. Mostly the topic are gastrointestinal physiology, intestinal mucus, diarrhea, and ion transport. I think that's all about Professor Kim Barrett, our distinguished lecturer this morning. I'm sure we will have a lot of new knowledge and information about the gastrointestinal system. We are lucky to have Prof. Kim Barrett for giving us a lecture. I hope students can use this opportunity to get the most updated knowledge about this topic. Once again, thank you very much, Professor Kim Barrett, for your time. I hope we will finish before midnight there. Thank you very much. Thank you, Dr. Lydia.
Thank you, Dr. Ermita. Ladies and gentlemen, and now we have to come the moment that we all have been waiting for. I would like to invite our honorable speaker, Prof. Kim Barrett, to deliver the lecture. Prof. Barrett will deliver her lecture in 50 minutes. And please, all participants are welcome to drop your message or question on the chat room by mentioning your name and your institution. When time permits, we will choose the best question to answer by Prof. Kim. Prof. Barrett, the stage is yours.
Thank you. And I will share my screen. Hopefully this will work.
Perfect, Prof.
Can you see the slides?
Yes.
Perfect. Thank you. Well, I want to thank you very much for the opportunity to present this lecture, and particularly Professor Amita, who extended the very kind invitation. Of course, I wish I was speaking with you in Indonesia right now, especially perhaps in Bali, but I am speaking to you from Washington, D.C. in the United States, and I'm very honored to have this opportunity to speak to you. I'm going to speak about a few remarks about gastrointestinal physiology generally, and then I've chosen a few topics of recent advances in the field that I think will be important facets of gastrointestinal physiology going forward. So many of the things that I will talk about really are still in the research realm, but they are the sorts of information that will be in the new textbooks in coming years. So the first thing I should say is I have been a student and a researcher of gastrointestinal physiology essentially my entire career since I started my faculty position in 1985. And I really love gastrointestinal physiology. And on many levels, it's not a very complicated field. We take in food, and the waste products exit at the other end. But as you all know, the situation is considerably more complicated than that. The key functions of the gastrointestinal system, of course, first and foremost, is to allow for digestion and absorption of nutrients so that they can be assimilated into multicellular organisms. And the gastrointestinal tract is also a very convenient route that we can use for therapy. So that's also very important to understand from a medical student perspective. So the vast majority of pharmaceuticals that are useful in the clinic are given through the oral route. So an understanding of gastrointestinal physiology also contributes to our understanding of pharmacology. The gastrointestinal system is also a key organ or system of excretion. And it is not only the residues of ingested food that can't be assimilated, or bacteria and cell debris, as we'll hear, but also we use our gastrointestinal tract to excrete predominantly lipophilic substances that cannot be excreted in the urine. And these are secreted into bile and then excreted through the gastrointestinal tract. And then another really key function of the gastrointestinal system, because it is essentially contiguous with the outside world, is host defense. And there are several aspects of that host defense. A very sort of basic aspect of host defense for the gastrointestinal system is the gastric acid that is produced by the stomach, which largely sterilizes the meals that we ingest. But the gastrointestinal system also has an extremely well-developed immune system. And in fact, it's been estimated that there are more immune cells in the gut than there are circulating in the circulating immune system. And there are sophisticated defenses from both the innate immune system, but also acquired mucosal immunity. And one of the really important facets of gastrointestinal physiology is how we can ingest a wide variety of potentially antigenic substances with impunity and yet mount appropriate immunologic responses to potentially harmful agents such as pathogens. So if we think about an overview of the gastrointestinal system, thinking of the system itself as a machine, the mouth we can consider to be a chopper that disperses the food that we take in into smaller pieces. That meal then traverses a transfer tube, also known as the esophagus, to enter the stomach compartment, which we can consider to have three main functions. It's like a blender that homogenizes the meal into a suspension of small particles with greatly increased surface area. As I already mentioned, it's a sterilizer by virtue of its ability to produce gastric acid. And it's also a reservoir by virtue of the property of receptive relaxation, by which the contents of the meal can be held in the stomach and then gradually delivered downstream to match delivery of nutrients to the digestive and absorptive capacity. In the small intestine, in the center of the figure, we can consider that to be a reaction vessel where the food that we take in is mixed with enzymes and other substances. Some of those come from the liver and the biliary system in the form of detergent or bile acids. Others come from the pancreas, shown on the right-hand side in the rectangle, both enzymes and also bicarbonate, which neutralizes the gastric acid. And then finally, the components of the meal travel through the remainder of the small intestine, which has both a catalytic surface that performs the final stages of digestion and an absorptive surface by which the assimilation of now small molecules derived from the food that we eat are absorbed into the body. And then the large intestine, which is depicted in this model as the circle at the bottom of the figure, is a combustor of the residues of the meal that our own enzymes are not able to fully digest, such that they're in an absorbable form, and particularly the combustion of dietary fiber into short-chain fatty acids that can be assimilated. The large intestinal colon also serves to desiccate the residues of the meal and to reclaim the large volumes of fluid that are used during the course of digestion, and also pellets the residues so that they can be conveniently expelled through the emission control device, also known as the anal sphincters. Another important aspect of the gastrointestinal system is its very significant surface area. Has been estimated to approximate the area of a tennis court. And this provides us with a very large reserve capacity for nutrient absorption. So nutrient absorption is extremely, we have a lot of capacity, and this probably was very useful in times of scarcity. But the large surface area can also be a liability in the setting of diarrheal diseases, because it is across this large surface area that diarrheal fluid can be produced. So in terms of general features of gastrointestinal physiology, there is extensive crosstalk between the various segments of the system, both feed-forward signaling, whereby the more distal parts of the GI tract are prepared to respond and to anticipate the components of a meal, but also feedback signaling. For example, signaling from the small intestine to the stomach to slow down the delivery of nutrients from the stomach. one of the aspects, I think, of GI physiology that is challenging for the beginning medical student is that there are so many different regulatory systems. And these multiple regulatory systems often also have redundant or overlapping functions. So it's difficult to understand the relative importance. As I already mentioned, we have excess capacity for digestion and absorption as well as extremely high efficiency. And this presumably provided an evolutionary advantage when our ancient ancestors really didn't have a ready or reliable access to nutrients. But in the modern world, and particularly in the United States, where obesity is a major public health issue, this is a liability at times of nutrient excess. The gut is also part of a very complex communication system with the brain, and we'll be talking a little more about that this morning. And there's also, not uniquely, but perhaps more extensively than anywhere else in the body, a really lifelong association with a complex microbial community, which recent years have taught us a great deal about, and I will be addressing in this presentation. So in terms of the topics that I want to cover this morning, first of all, I'm going to talk about the enteric nervous system, the so-called little brain of the gut, and its role in diseases such as inflammatory bowel diseases and irritable bowel syndrome. I want to talk about bile acids, which formerly we simply considered to be biological detergents that helped us to digest and absorb lipids, but we now really recognize them as so-called enterohormones, substances that are important in regulating the function of the gastrointestinal tract from within the gastrointestinal lumen. I'm going to briefly cover some emerging information on the ways in which the gut handles and responds to dietary lipids. And then finally, I'll touch on emerging roles of the gut microbiota. So moving first to the enteric nervous system. The enteric nervous system is often referred to as the little brain. And survival of multicellular organisms such as humans definitely requires the integration of the motility functions of the gut that move the contents of the meal along the length of the bowel, the secretory functions that mix the food or the chyme with gastrointestinal secretions to allow for digestion and absorption, and also blood flow to carry away digestive nutrients. And that trio of key gastrointestinal functions that are essential for survival are regulated by the enteric nervous system, which is a complex network of neurons and also glial cells that are predominantly arranged in two plexuses along the length of the bowel. The myenteric plexus that is sandwiched between the longitudinal and circular muscle layers of the gut, and the submucosal plexus closer to the mucosal surface and the gut lumen, which is almost certainly predominantly involved in sensing the minute-to-minute composition of the gut contents and generating appropriate physiological responses to the physicochemical characteristics of the bowel. The little brain of the gut communicates with the central nervous system, but it's also largely autonomous of the central nervous system, and that's why it was referred to as the little brain. It can mediate stereotypical responses of GI physiology entirely independent of any central nervous system involvement. But as we all know, the central nervous system can also affect our gut function, and stress, for example, or other systemic physiological imperatives will definitely have an impact on the function of the bowel. And developmental defects in this little brain, as well as acquired defects in enteric nervous system signaling, may underlie a variety of bowel disorders. So thinking about these two plexuses of that comprise the majority of the enteric nervous system, as I mentioned, the enteric plexus is between the longitudinal and the circular muscle layers and is largely concerned with regulating both the contraction and the relaxation of those muscle layers to move the gut contents along the length of the gut. The submucosal plexus is located between the circular muscle and the lamina propria of the gut. It regulates fluid secretion and absorption and also blood flow, and it responds to stimuli coming from the epithelium and from the bowel lumen itself. And that also includes responses to substances produced by the microbiota that lives within the bowel lumen. So a lot of recent work has emphasized and has uncovered important facets of how the enteric nervous system develops. So we now know that in embryological terms, cells that are derived from the neural crest migrate through the mesoderm and then move in a rostral to caudal direction to colonize the fetal bowel at the level of what will eventually become the myenteric plexus. So the nerve cells both proliferate and then migrate along the length of the bowel from the anus towards the head. A subset of these cells responding to specific signals within the gut wall will then migrate radially away from the myenteric plexus and towards the lumen to form the submucosal plexus. And then much as we see in the central nervous system, where different functions are produced by different types of nerves, we now know that there are at least 20 different neuronal subtypes within the enteric nervous system, plus the enteric glia that regulate neural function. And these subtypes have different functions. They contain different neurotransmitters. They have different patterns of their neurite projections, and they also have different electrophysiological characteristics. And it has not been precisely mapped to specific functions, although much of this work is ongoing right now. But presumably, this heterogeneity of the enteric nerves underlies the regulation of the stereotypical motility and secretory patterns of the bowel. For example, peristalsis or segmental motility patterns, segmentation that mixes the bowel contents, or reflex secretory responses as the gut contents pass along the length of the epithelium. So to show this in a more diagrammatic fashion, a lot of the specific factors have been uncovered that regulate the development of the enteric nervous system. And we also know that defects in both the signals and the receptors for the signals can result in defects in development of the enteric nervous system. So initial precursors in the neural crest migrate through the mesoderm, where under the influence of retinoic acid, they acquire expression of RET receptors, which are receptors for glial-derived neurotrophic factor that stimulates the proliferation of these initial precursors. And typically, these precursors will then go on to migrate, as I mentioned, and to sort of get cues from the non-neuronal cells to which they adhere, and they migrate rostrally to caudally to produce all of the differentiated cell types of the myenteric plexus. If these initial proliferating precursors encounter signaling from the notch pathway, they will differentiate along a separate pathway and form the glial cells. And then ultimately, when the cells reach their destinations in what will become the myenteric plexus, they are acted on by another factor called netrin as they acquire expression of DCC, which stands for deleted in colon cancer, which is the netrin receptor. So netrin is the factor that drives that radial migration of the nerves to produce the submucosal plexus. And it's also involved in migration of sensory nerves arising from the vagus to innervate the bowel and to sense bowel function. So as migration nears completion, the neurons aggregate into the ganglia that characterize the mature enteric nervous system. And then in those ganglia, they terminally differentiate into the different neuronal subtype. There's probably a role for a balance between GDNF, as I mentioned previously, which also triggers migration and proliferation initially, but also bone morphogenic protein, which causes differentiation rather than proliferation. And also endothelin-3 is known to be a very important signal for migration, but cessation of that signal is a trigger for terminal differentiation to begin. The neurons then, once they're in position, they start to extend processes to establish the circuitry in the enteric nervous system and allows the nerves to communicate. That is, again, very important for their function to regulate motility and secretion of the bowel as a whole. And then this is not all completed within embryonic life. Development of synapses, refinement of the neural network and remodeling of the network continues postnatally, both in health, but also can be interrupted or disrupted by specific triggers. So for example, if there are early life environmental disturbances that may impair the final complete maturation of the enteric nervous system and lead to issues with motility and secretion later in life. We also have recognized in recent years that the microbiota is an important regulator of the enteric nervous system. So in fetal life, it's widely held that the gut is sterile, but almost immediately upon birth, the gut begins to be colonized by a complex microbiota that then matures over the first few years of life. We know from experiments in mice that have been raised to have no microbiota, so-called germ-free mice, their gut function is abnormal. Their motility is altered, and they have a decreased density of the enteric nervous system. So a sort of defect overall in the ability to regulate the bowel. It's also known in adult life that if you modify your diet, and probably all of us have experience of this, it can change transit time in the bowel in concert with effects on the microbiota, which is correlation, but suggestive of the fact that the microbiota might in fact be regulating transit by regulating the enteric nervous system. We know that some of these effects of microbes and their metabolites are direct, but some of them are likely secondary to activation of immune cells and or epithelial cells. And it's also likely that the communication between the microbiome and the enteric nervous system is bidirectional, because in Hirschsprung's disease, where there is a failure of the neuronal progenitors to migrate fully throughout the length of the bowel, the microbiota was altered. So that may be secondary to altered motility, or perhaps is directly reflective of interactions between the nerves and the microbiota itself. And work from Vasilis Paknis's lab in London has also suggested that the microbiota actually drives the appropriate postnatal development, the final stages of development of the enteric nervous system. And they have identified macrophages residing in the lamina propria as a cell type, an immune cell type that responds to microbes and their products, produces factors such as bone morphogenic protein that causes final maturation of enteric neurons. Those neurons in turn can produce growth factors such as colony stimulating factor 1 that can act on the macrophage. And similarly, as yet less well defined, but there also appears to be crosstalk between macrophages and precursors of enteric glial cells. So this remodeling continues throughout life. So what about plasticity of this system in particular disease states? So there's definitely an interaction between the enteric nervous system and inflammation, and environmental influences and disease may induce changes in either the coding of the nerves or their function, even in adulthood. We'll talk a bit about so-called post-infectious irritable bowel syndrome with predominant diarrheal symptoms, post-infectious IBS, and in North America that has taught us a lot about how that syndrome arises. And then within both patients with inflammatory bowel disease or in models of colitis, there are many changes that have been described in the enteric neural circuits, and they may be responsible for aberrant motility and pain and diarrheal symptoms seen in patients with inflammatory bowel disease. So the Walkerton outbreak was a set of gastroenteritis cases that occurred in a small town in Ontario, Canada, called Walkerton. There was contamination of the town's water system by fecal material from cattle. There was a breakdown in the sewage system, and this basically contaminated the town's water supply. And over 2,000 people got sick with a pathogenic E. coli, and 7 people actually died. Now, this would be just a sort of typical story about an outbreak of infectious diarrhea. But the most interesting thing perhaps was that within 2 years of this outbreak, it was noticed that more than a third of the patients who had had and recovered from the initial waterborne illness had symptoms of irritable bowel syndrome with remitting and relapsing episodes of diarrhea. Women, children, and those who had been the most seriously affected initially were at greater risk of developing these symptoms. Now, this has happened a number of years ago now, and at 10 years of follow-up, most people had spontaneously recovered, which is a good thing. And it's felt that the functional bowel disease that arose in these individuals that could be studied in great detail essentially reflected a crosstalk between the enteric nervous system, the epithelium, and a decrease in barrier function, and therefore altered both motility and visceral sensation. And some of this is also summarized in this cartoon here from a recent review article that shows down at the bottom at point 1 that epithelial cells are influenced in the setting of inflammation because serotonin or 5-hydroxytryptamine is released in the vicinity of sensory afferent nerve fibers. There's also death of some of the neurons in the setting of inflammation, and others may become hyperexcitable and result in increased synaptic input. But there are also, in terms of the circuitry that normally controls the orderly patterns of gut motility, there may be increased synaptic potentials, both in excitatory motor neurons, which cause contraction of the circular muscle, and also in the inhibitory motor neurons that cause relaxation. And there's also decreases in specific classes of neurons that mediate purinergic transmission. And so based on studies in humans with inflammatory bowel disease and also in animal models, on the upper part of this figure, you can see what happens in healthy individuals when there is a trigger for peristalsis, a distension of the bowel. That will be a trigger for contraction behind the bolus, shown by the green neurons. And that is mediated by activation of the sensory neurons where the bolus is found, and relaxation mediated by triggering of the inhibitory motor neurons so that there can be an orderly progression. But in the setting of inflammation, the pattern of signals is disrupted. There is a mixture of signals, and this interrupts propulsive motility and is almost certainly part of the symptom complex that accompanies inflammatory bowel diseases. So to pull these into some clinical correlations, as I already mentioned, Hirschsprung disease that is typically identified in infants reflects an incomplete population of neurons and ganglia in the gut due to defects in either proliferation or migration. That could include mutations in the RET receptor for GDNF, or the endothelin receptor, or the ligands for those receptors. In irritable bowel syndrome, it appears that there are more subtle changes in the development and differentiation of the enteric nervous system, but we definitely need additional study. But the possibility that sort of plasticity of the enteric nervous system in adulthood can lead to disease is definitely underscored by post-infectious IBS, such as was seen in Walkerton. And then in inflammatory bowel disease, neuroplasticity that is induced by bowel inflammation disrupts propulsive motility. There's also impairment of the gastrocolic reflex that would normally retard transfer of meal from the stomach to downstream segments of the bowel, so that can also contribute to diarrheal symptoms. There's also a lack of retrograde colonic contractions that normally retains the residues of the meal in the colon for a sufficient period that they can be dehydrated. And then we know that some of these changes in nerve function can be long-lasting. So I'm going to move on now to talk about bile acids as so-called enterohormones. We have known for many years, and I'm sure that the medical students in the audience have certainly understood, that bile acids that are produced by the liver are very important to solubilize the products of fat digestion in the aqueous environment of the gut lumen. And the micelles that are produced by interaction of these amphipathic bile acids with lipids allow us to more efficiently absorb the products of lipid digestion, such as monoglycerides and free fatty acids. And these micelles, mixed micelles that include bile acids, are absolutely required for the absorption of fat-soluble vitamins and cholesterol. So in the absence of sufficient bile production, you can definitely become deficient in fat-soluble vitamins. We do know that bile acids circulate in the bloodstream in addition to their large reservoir in the bowel lumen. And that's comparable to other GI hormones. They circulate and they fluctuate with meals. But in addition to their sort of detergent properties, in the last 10 years or so, it has bile recognized that these are not just inert detergents, they actually are signaling molecules. And 2 receptors have been identified that mediate bile acid effects. The farnesoid X receptor, which is a nuclear receptor, and a receptor called TGR5, also known as GPBA for G protein coupled bile acid receptor, that is expressed on the cell surface of a variety of different cells. And changes in bile acid signaling and bile acid handling have been implicated in various digestive and other disease states. So you're probably all aware that bile acids circulate. They are produced by the liver. They're stored in the gallbladder. They enter the gastrointestinal lumen. They travel through the small intestine. And then and the terminal ileum, they are largely reabsorbed. But they can move into the colon, where a portion will be lost in the feces, but most will, that spill over from the small intestine will also be reabsorbed, travel through the portal vein back to the liver to recycle. So we have this daily circulation of bile acids. And these bile acids are produced from cholesterol by hepatocytes into 2 primary bile acids, chenodeoxycholic acid and cholic acid. And then those bile acids that make it to the large intestine are acted upon by colonic bacteria to produce the secondary bile acids of deoxycholic acid, lithocholic acid, and ursodeoxycholic acid. And then those bile acids can be reabsorbed back into the portal circulation, either by active transport in association with sodium, shown in the upper green cell, or passive diffusion through the epithelium. And most of the bile acids return to the liver and are resecreted back into the bile. But as I mentioned, there is some spillover of bile acid species into the systemic circulation. I'm not going to go into this slide in great detail, but just to alert you to the fact that just about any cell type that you can think about that might be involved in gastrointestinal function, or indeed in other functions such as immune reactions and whole body energy metabolism, respond to bile acids via this cell surface receptor TGR5. There can be gallbladder epithelial secretion or muscle reaction. There's activation of enteric nerves, activation of cells that produce enteric hormones. So they clearly have very wide-ranging activities through this cell surface receptor. And because of these wide-ranging activities, when there is abnormal bile acid signaling, there may be a disease that results. So for example, for irritable bowel syndrome with predominant diarrhea, it has been estimated that as many as 1/3 of the cases of that syndrome may in fact represent bile acid diarrhea. And there may be also genetic variations in the TGR5 receptor that can contribute to changes in motility. In cancer, deoxycholic acid has been implicated in driving the growth of both gastric and esophageal cancer cell proliferation. High levels of bile acids can be injurious to the liver, causing apoptosis of the hepatocytes. On the other hand, low levels of bile acids that are seen in health are anti-inflammatory. And then bile acids signaling through TGR5 do contribute to mitigating the effects of a high-fat diet and protecting against metabolic disorders and obesity. And in part, these effects of bile acids have been implicated in the beneficial effects of bariatric surgery for those who are seriously obese. Bile acids also participate in important feedback loops that regulate their own production. So on the left-hand side of this slide is the situation in health, where bile acids are produced by the hepatocytes. In the upper part of the slide, they travel through the gut. They are absorbed by the epithelial cells in the ileum. And in those epithelial cells, they interact with the FXR receptor, which triggers the synthesis of a hormone called fibroblast growth factor 19. FGF19 travels through the portal circulation to the liver, where it interacts with its receptor and an accessory protein called beta-klotho, and that reduces bile acid synthesis. So as this travels back, if bile acid levels rise, then synthetic capacity will be reduced. However, in bile acid diarrhea, there has been identified mutations in the receptor for FGF19. There are also the possibility of impaired absorption of bile acids in the small intestinal lumen, which again will reduce FGF19 production because there's less bile acids to interact with FXR. And the net effect of disruption of these feedback loops is an increase in bile acid synthesis. Those bile acids, moreover, if they are not absorbed by the ileal epithelial cells, will travel into the colon, where they can increase colonic motility, increase visceral sensation and pain, increase fluid secretion, and increase mucosal permeability, mostly by acting with the cell surface receptor, TGR5, and that then can contribute to bile acid diarrhea. So a few words about the handling of dietary lipids. We know that dietary lipids play a central role in energy homeostasis. They're important nutrients because they're calorically dense. And we also need lipids in our diet for the synthesis of our cell membranes and for various lipid mediators. Our body has evolved to carefully monitor fat intake and to optimize sensing and storage of lipids. And the availability of fat-rich foods varies by season and by where in the world you happen to be. And we know a lot about digestion and absorption of water-soluble nutrients, such as carbohydrates and proteins, but the understanding of digestion of lipids was much slower to emerge, but there has been several recent advances. Now, this is a complicated slide, but I think I just want to stress a few items on it. So in the upper part of the figure, you can see that dietary fat is acted upon, formed into mixed micelles with the action of bile acids, acted upon by various lipolytic enzymes, including those coming from the pancreas. And we now know that the free fatty acids and the monoacylglycerol that is produced by this enzymatic activity, which were previously thought to diffuse passively into the epithelium, in fact is mediated by a protein called CD36. I'm not going to get into the details of chylomicron formation, but I want to talk about a couple of other transporters on the epithelial surface. One of them, shown here as NPC1 like L1, stands for Niemann-Pick C1 like 1 protein, which is a transport protein for free cholesterol. And so that is a potential pharmacological target by which absorption of both dietary and excreted cholesterol could be interrupted. We also know that absorption of cholesterol from the gut lumen is relatively inefficient because it is also actively effluxed from the cells, as shown on the right-hand side, by an ABC transporter pair, ABC G5 and C8, and that active transport limits the reabsorption of cholesterol and allows it to be excreted from the body. So that is another potential pharmacological target. So we can actually use the gut to approach a systemic disease of hypercholesterolemia. We know that statins are very effective in reducing cholesterol synthesis, but perhaps because they reduce synthesis, they actually upregulate cholesterol absorption from the gut. So the gut is trying to hold on to cholesterol when it senses that synthesis is reduced. And a drug known as ezetimibe actually acts as an inhibitor of NPC1L1 to reduce the uptake of either dietary cholesterol or cholesterol secreted by the liver. So if you add this drug to statin pharmacotherapy, it can reduce circulating cholesterol in patients with both primary and familial hypercholesterolemia. And then also strategies whereby drugs would increase expression of these ABC transporters, and metformin has been shown to do this, could also be of benefit in diseases of cholesterol excess, because this would also further enhance cholesterol export. I think I'm going to skip that one in the interest of time. So finally, I just want to talk briefly about the gut microbiota and its impacts in health and disease. We know now that the gut is, and we've really known from a very long time, that the gut is far from sterile. It is the home to large numbers of microorganisms, not only the bacteria that are listed here, but also yeast and viruses and archaea. The composition of the gut microbiota varies along the length of the gut, and also the numbers of microorganisms. There's relatively few microorganisms in the stomach and the distal small intestine, but as you move distally, the density of microbial colonization is much enhanced. And that is mediated both by intrinsic factors such as gastric acid or the availability of oxygen or the motility functions of the gut, but also by both drugs, laxatives, antibiotics, and dietary factors as well. So motility influences gut microbiota, the availability of acid, and the availability of oxygen, such that in the small intestine we have predominantly anaerobic or facultative anaerobic bacteria, whereas in the colon we have strict anaerobes. So there's some debate about the precise number of bacteria and genes that are expressed in the colon, but it's almost certainly the case that there are more bacterial cells and or genes than there are cells and genes in the human body. They're an important metabolic factory, and we can ask the question, which is them and which is us? The gut microbiota is a central component of the host phenotype. Host factors, dietary factors, pharmacologic treatments, all of these will impact the precise makeup of the mixture of microorganisms. And that in turn will influence the ability of the gut microbiota to produce metabolites that can then act on the host and can result in changes in the host phenotype, such as fat mass development, glucose tolerance, inflammation, and the efficiency with which we can obtain energy from the diet. In essence, the host is shaping the microbiota by factors coming from immune cells, from the epithelium. The epithelial cells are regulating the mucosal immune cells that will shape mucosal immunity. And so there's this complex communication that's going on at all times that regulates the precise components of the microbiota. And from the epithelial perspective, we normally have a balance such that the barrier function of the epithelium allows the transmission of appropriate signals from those microbes present in our gut to the host. But if we have the loss of an effective barrier or the wrong mix of bacteria, then we get inappropriate signals from the commensal to the host immune system and perhaps perpetuation of inflammation. So in terms of clinical correlations, we know that the microbiota can protect against pathogens by direct exclusion and by having injurious effects to pathogens. There have been changes in the microbiota, so-called dysbiosis, that have been described in numerous digestive and extraintestinal disease states. There is a growing appreciation of the ways in which the brain, the gut, and the microbiota all talk to each other, and possible roles have been uncovered for the gut microbiota in a variety of central disorders such as anxiety, depression, autism, and as we spoke about, development of the enteric nervous system, but perhaps also development of the central nervous system. I will say there's one caveat here, which is most of the mechanistic studies that have been conducted have been in animal models, and the human studies have mostly been descriptive, but that is changing rapidly. So how does the microbiota signal to the central nervous system? It can do so by directly stimulating vagal afferents, by releasing metabolites or inducing the host to release metabolites that can travel to the brain through the circulatory system, and also by releasing or producing molecules that activate the immune system that again may produce cytokines or may travel directly to the central nervous system. So in the setting of a disease associated with dysbiosis, what can we do about it? Well, one possibility is to alter the diet, such as by delivering so-called prebiotics, which trigger the increased growth of presumed beneficial species. Or we can use so-called probiotics, which is by directly delivering friendly bacterial species to bolster their populations in the gut if they have been reduced by disease. And then also there is the possibility of directly transplanting the fecal microbes from a healthy individual to one with disease. In terms of the diet, prebiotics are fermentable substances. They can change the balance of the types of bacteria in the gut to produce different short-chain fatty acids that may or may not trigger the release of inflammatory cytokines. Other bacterial species, particularly in a high-fat diet, can produce products that are injurious to the epithelial barrier. And as you can imagine, this is a very complex system to work out, but some of the really outstanding metabolomic studies and very detailed bioinformatics are now trying to bring some order to this large amount of data. Probiotics have been shown to improve barrier function, to improve the resistance of the gut to colonization by pathogens. They themselves can produce antimicrobial products. They can stimulate mucosal immunity, and they may have anti-inflammatory effects. And then microbial transplantation refers to endoscopic delivery, or if you go online and Google, you can figure out how to do this yourself. And some people do try to do this themselves. And it's typically minimally processed stool from a healthy family member with the goal to restore a beneficial microbial community. Now, this seems like a very primitive medical treatment, but in fact, it's been shown to be dramatically effective in some intestinal diseases, particularly intractable infections with Clostridium difficile, which often overgrows and is very difficult to eradicate in patients who've had long courses of broad spectrum antibiotics. We really don't have such good data to suggest its efficacy in other GI conditions, although studies are ongoing. And the long-term goal is to figure out what is it in this stool that's being transferred whether it's metabolites or specific bacterial species, so that we no longer actually have to transfer the stool itself. So just to summarize, I've talked about the enteric nervous system and its role in intestinal disease, bile acids as possible hormones in the gut, how we handle and how we respond to dietary lipids in the bowel, and emerging roles of the gut microbiota. And I believe that we have had particularly rapid advances in the last decade or so in our understanding of many different facets of GI physiology, and I only had time this morning to touch upon a few. Ultimately, these advances have the potential to impact the treatment of digestive and other diseases that are impacted by the gut. And in turn, as we discover new therapies, that then takes us back to further illuminate the underlying physiology. So I thank you very much for your attention. I'm happy to take any questions that people may have, and I'm also happy to move to the question to the students part of the program.
Wow, what a very interesting and insightful speech it was. We learned so much about the new development of gastrointestinal physiology, Prof. Kim. Thank you, Prof. Kim Barrett, for your comprehensive elaboration. Ladies and gentlemen, let us give a round virtual applause for Prof. Kim. Thank you, Prof. Kim.
Thank you.
Professor, ladies and gentlemen, now we will move on to special question and answer session. Usually, the audience will give question to the speakers. However, in this session, Prof. Barrett has already prepared for set of questions for the student. These students will try answering the question. Later, Prof. Barrett will provide a thorough explanation regarding the answer of the questions. Each of us can also try to answer, silently of course. Each of question and answer session consists of 3 questions and will last for around 8 minutes only, and is led by a host who is a lecturer for their university. And our IT staff will share the question on screen, and we can read it for 30 seconds. Any student who knows the answer can use the raise hand feature on Zoom, so the host will choose and invite him to answer. Student, important, student who gives the best answer or explanation from each session will get a prize from our sponsor, Elsevier Publisher. Now we will start the first set of questions. This set will be given to students from Muhammadiyah University of North Sumatra, Medan. I invite Dr. Debbie Lubis to host this session. Your time is 8 minutes.
Okay, thank you very much, Dr. Widya. Okay, thank you for Prof. Kim, especially. Good evening. Students from University Muhammadiyah of North Sumatra, maybe now you can see 3 questions on screen, and you can read carefully, and then I will choose 3 of you to answer the question. Okay, maybe we start from question number 1. Okay, you can have 30 seconds to read the question first. And now I will ask to Bella Nurhalida. Can you hear me, Bella?
Yes.
Okay, can you answer the question number 1?
Yes, I can.
Please.
The method to culture gastrointestinal microbiota is challenging and has limitation, but the limitation has been partially overcome by molecular approach such as DNA sequencing, which target of this technique is 16 as ribosomal R antigen. That is also useful for tracking temporal change or perturbation in response to antibiotics. The example is cancer. Cancer can be related to the decrease in diversity and community stability of gut microbiota. like the bacterial RNA sequencing reveals reduced levels of firmicutes in colorectal cancer patients. And and the inflammatory bowel show the example that with advance in DNA sequencing indicated that microbiota function was consistently more perturbed than composition, that leading to the conclusion that microbiota composition studies may underestimate its impact on IBD pathogenesis.
Okay, thank you very much. And now maybe I ask to Prof. Kim to review the answer. Please, Prof. Kim.
Yes, that's really nice. And thank you very much for your very complete answer. So definitely one of the things that we can do is just go and look and see what's there and say, if you have people with a disease, what is their microbes compared with somebody else? But the other issue is that is a correlation, not a causality. And some of the things that have been done to look for causality is to take the microbes from, for example, a patient with a specific disease or a specific condition and give those to a germ-free animal compared with the normal microbiota. And I think the most convincing experiment that was done in this regard was microbiomes were collected from obese subjects versus lean. And when they put the microbiome from an obese human subject into a germ-free mouse, that mouse became obese, whereas from a lean individual, they stayed lean. So that is a more causative experiment, but of course requires somebody to do this in the lab in very specialized equipment. But you did a very nice job of answering the question.
Okay, thank you.
Excuse me, Dr. Devi.
Okay.
May I interrupt? Could we see the participants that's answering? Could they put on their video so we can see their faces? Thank you.
Yes, thank you, Dr. Minarma. Yes, for all students that I choose, maybe you can turn on your video before I answer the question. Okay, we will go to question number 2. Okay, I ask to Muhammad Dafa. Can you answer the question number 2, Muhammad Dafa? Muhammad Dafa Pratista from UMSU. Can you hear me? Please on your video and audio. Muhammad Dafa?
Yes, ma'am. I'm sorry, there's a problem.
Oh, okay, okay. Thank you.
Yeah, number 2. Yeah, thanks for the opportunity. Before, yeah, I am the number 3. What approach can be used in human to modify the constitution of the microbiome, either temporarily or over the sustained period of time? Okay, there are several approaches that can be used by human to modify the constitution of microbiome. They include lifestyle factors such as carbohydrate and fibrous diet, caloric restrictions, and next there's exercise, cigarette smoking, and alcohol consumption. But in other hand, we can use clinical intervention such as fecal microbiota transfer, using antibiotics, probiotics, prebiotics, and
Very noisy there. Yes, Dafa. Maybe now I will let Prof to review the thank you, Dafa.
I'm sorry, ma'am. I can hear you.
Okay, thank you very much for the answer. I will let Prof Kim to review the answer. Please, Prof Kim.
Yeah, very excellent set of suggestions. In fact, it's actually quite difficult. We do know that the microbiome is dynamic. It changes even during the course of a day. But in any given individual, you can perturb it temporarily, but as soon as you stop treating, then it may well go back to its previous state. But certainly antibiotics and long-term changes in diet will have a significant impact. But thank you for your very comprehensive answer.
Okay,
Thank you.
thank you. Thank you, Prof. Kim. Okay, the last question. Maybe I will ask Hairunisa. Hairunisa Karim,
Yes, ma'am.
can you hear me? Yes,
Yes.
please answer the question number 3.
Okay, thank you. Thanks for the opportunity.
Your video and audio, please.
Okay. Thank you for opportunity. So I would like to answer this question. There are several symptoms that caused by broad-spectrum antibiotics. Those are diarrhea, abdominal pain, stomach cramp, and vomiting and nausea. It can be found in patients with colticioid membrane. It occurs due to long-term use of antibiotics that can lead to colonization of normal flora, such as Clostridium difficile, and become pathogens. Since this bacteria multiply, it can produce toxins, and if the human response is inadequate, it can cause colitis. Thank you.
Okay, how about the answer, Prof. Kim, please?
Yes, that's really excellent. And I don't know about the situation in Indonesia, but this is a really major problem in the United States, is this C. difficile colitis that is produced by the use of antibiotics a lot of the time in hospital settings, but people are really made very sick by this bacterium because of the toxins that it produces. So very nice job with the answer.
Okay, thank you very much, Prof. Kim and my 3 students here, and I will give the forum back to Dr. Widya. Please, Dr. Widya, thank you very much. Audionya Dr. Widya belum nyala.
Okay. Thank you, Prof. Barrett, for the explanation. And also thank you, Dr. Lubis, for the hosting this session. And especially for Bella, Dava, and Haurunisa. Thank you for your very clear answer. Now we will start the second set. We will share the question on screen. This set of questions will be given to students from University of Pajajaran, Bandung. I invite Dr. Yuni Susanti Pratiwi to lead this session. Your time is at minute, Dr. Yuni.
Yeah, thank you, Ms. Moderator, for this opportunity, and also thank you, Prof. Kim Barrett, to also observing our student how they can answer the question. So for all the participants, you can see the question on the screen, and maybe you can all just train yourself how to answer this question. Especially for my student, Rafaela, Bistami, and Afif, please make sure that you can read the question, and also please make sure that you on the mic and also turn on the video. Okay, so let's start with the question number 1, and I will let Rafaela. Rafaela, can you hear me?
Okay, I can do that. So let me answer the first question.
Oh, you just directly want to answer? Okay. Okay, just please go, Rafaela.
Okay, to begin with, from hepatocyte, cholesterol is converted into the cholic acid, and this acid combined with glycine and taurine to form glycol and tauro-conjugated bile acid. Also, there are salts of this acid that composed from mainly sodium salts and then secreted into the bile. Bile acid and bile salts are secreted into bile canaliculi, which originate from between hepatic cells via canals of Herring, and then bile flows toward the interlobar septa and empty into the terminal ducts into larger ducts, and finally reaching the hepatic ducts and common bile duct. And from this duct, the bile empties directly into the duodenum when meals are taken as a response to secretin and cholecytokine that released by duodenum, esophageal stimulation response, and acidic content that contact with duodenum, which leaving stomach, that causes the gallbladder contract and the sphincter of Oddi relaxes, and facilitating the bile flow into small intestine, and in the intestine, bile acids from micelles. Those bile acids mostly reabsorb reabsorbed by ileum epithelial cells and to the hepatic portal vein, and about 5% are secreted into the feces. On the other hand, during the fasting state, bile acid is diverted into the cystic duct into the gallbladder and stored in the gallbladder during fasting state, and the epithelium of gallbladder will absorb the water and electrolyte through the action of membrane channels and transport. That's all my answer. Thank you, Doc.
Thank you. How about that, Prof. Kim? Yeah, please.
That was fantastic. Well done, Rafaela. I mean, really nice. I think the only thing that you maybe you said it and it went by very quickly, but there is some spillover of the colic acid into the systemic circulation as well. So if you could see rises in the systemic circulation and then falls after the meals. But you had a really fantastic answer. So congratulations.
Congrats, Rafa, for those speedy questions. Okay, next question to Bistami. So Bistami can also open mic and also open your video. You have 30 seconds to prepare yourself, or you just want to answer directly now?
Yeah, I will answer the question, Doctor. First of all, thank you to Dr. Yuni, Dr. Widya, and Professor Kim Barrett for this opportunity. So colic acid is one of the main components of bile acid, which in turn is a component of bile, which comprise of other components such as bilirubin. The first step by which it is synthesized is through the conversion of cholesterol, which is derived from the fat that we eat by the enzyme called 7-alpha hydroxylase in the smooth ER. And eventually, after several steps, it will produce the so-called first bile acid, which is either quinodeoxycholic acid or cholic acid, which is either, and those two things can be further metabolized through the conjugation with taurine or glycine. Then the cholic acid or the derivative from it will be secreted to the intestine to emulsify the lipid. Most of the time, the bile acid will undergo a circulation back to the liver to be converted to the second bile acid in order to make it more polar. Then the second bile acid will be secreted back to the intestine. That's all my answer, Doctor.
Thank you.
So, I'm sorry, Professor.
Yes, please, Prof. Kim Barrett.
So, where do the secondary bile acids get formed?
It is formed with the help of the bacteria in the intestine. Yes.
Yes, so they're definitely being formed in the large intestine, and they can be passively reabsorbed. So they will also travel back to the liver. But yes, you did a very nice job. And knowing about the conjugation, which also reduces passive reabsorption, is also very important.
Thank you so much, Professor.
Thank you so much, Professor, for your constructive comments. And this is the last question for this session. I will call Afif. Afif, can you open your mic and also the video?
Yeah.
Okay, you can start answering now.
Yeah, I'm not really sure what the answer is, but I will try to answer it directly. I think there might be some protein, some kind of protein which function as a transporter to help this cholic acid energy drive to travel from one side to another side. From an article I read before, there's protein, there's transporter called BSEP transporter, or stands for bile salt export pump, to help cholic acid energy drive to do this function, to do this traverse through cell membrane. I think that's all.
Okay, thank you, Prof. Kim. Maybe you can give us explanatory.
Yeah, so the bile salt export pump is very important in producing the bile in the first place, so exporting the bile acids into the bile. Another one that's probably important is there is a sodium coupled bile acid transporter in the terminal ileum, and this is responsible for the active reabsorption of conjugated bile acids so that they can be recycled through the portal vein back to the liver, and so to enter this circulation through the enterohepatic circulation. And we know that in some patients that ileal sodium coupled bile acid transporter is mutated, and that can lead to bile acid diarrhea because you fail to reabsorb the bile acid from the small intestine, and then you have excessive concentrations in the colon. But thank you.
Thank you, Prof. Kim. I think this is the end of the session, but I would like to say something about the session. So I just purposefully choose the student from second semester, Prof. So they are not yet have a gastrointestinal lecture. So just challenge them to just following your lecture for the first time and just try to answer it. And they have a really awesome comment about the lecture. Thank you, Prof. Kim. Thank you.
They did a great job. All the students have been fantastic. So
thank you. So I will give back the forum to the moderator. Thank you.
Thank you, Dr. Yuni Susanti, for leading this session, and also thank you to Rafaela, Bustami, and Afif for the extensive answer for fate of cholic acid in this session. Now, the third set of questions will be given to the student of Hang Tuah, Surabaya. I invite Dr. Dodi Taruna to lead this session. Your time is 8 minutes, and please share the question on screen. Mbak Aulia, okay, already there. Please, Dr. Dodi.
Thank you, Dr. Widya. Good evening, Prof. Barrett. Okay. For the first question, I'd like to ask Calvin Juso to answer. Calvin?
Hello?
Yes, Calvin. You can answer the first question.
Okay, so hello, Professor Barrett. My name is Calvin. So first of all, because of the symptoms, we've seen that here it says, so this woman has come from Brazil eating a snack, recovers, so has taken medicine and came back to the United States and then has recurring episodes of abdominal pain. Which come and go over the year. So for differential diagnosis, we've actually thought of things or diseases like Crohn's disease. However, we have come up with the diagnosis of irritable bowel syndrome, just because through symptoms, obviously it is a little more difficult because we would want laboratory exams of diarrhea to rule out, for example, Crohn's disease with blood and stool. But yeah, for the diagnosis that we have come up with for 100% would be IBS or irritable bowel syndrome.
Nice, very nice. Yeah. So this is a classic presentation of post-infectious irritable bowel syndrome. So it typically is diarrhea predominant irritable bowel, and as you know, I'm sure from your reading and sort of digging into this, that there's very little in terms of evidence of any specific organic disease or major inflammation, and a lot of the time these patients have extremely extended workups to try and figure out what's going on. And sometimes people don't remember that they had an acute, what was presumably infectious diarrhea until a lot of questions. So nice job. Thank you.
Thank you.
Thank you, Prof. Barrett. And the second question, Anmal, please, would you please answer the question?
Okay, thank you, Dr. Dodi. Good morning, Prof. Barrett. Can you hear me?
Yes.
Okay, so thank you for the chance of us answering this question. So I would like to answer the mechanism that could happen to this woman. So my friend said that this is irritable bowel syndrome, and irritable bowel syndrome itself is an unknown disease. We don't really know what the etiology of this disease. So we were thinking about that there's the change in her bowel habit when she was traveling in Brazil, and that change of habit alter her persistent change in her mucosal immunocytes, enterochromaffin, muscles, anthrax nerves, and gastrointestinal microbiota. But I would also underline that there is other things that would happen, because in this case, we are also looking at the systemic thing, and it is the the 5-HT, or the receptor that you mentioned in your presentation, and it controls the motility of the gastrointestinal. So I would say that there's an alter in the receptor or the transporter of that hormone that could alter her motility, that causes her having diarrhea all over the year.
Yeah, very nice answer. And in addition, you know, the part of the etiology of the disease is thought to be a recoding of the enteric nerves, in part so that they are sensitized to stimuli that would normally be innocuous. So if there's any bowel distension, then this will activate the sensory nerves, and again could cause the release from the enterochromaffin cells of excessive amounts of 5-HT. So there's probably multiple mechanisms acting in concert, but very nice job. Thank you.
Thank you, Prof. Barrett. Thank you, Anal. And the next question for Rahaf. Please answer the third question.
Yes. Okay. First of all, I'm very grateful for being given the opportunity to answer a question from Professor Kim Barrett. Let me introduce myself. My name is Rahaf from the Hang Tuas University Faculty of Dentistry. The answer to your question is, if diarrhea is a symptom in PIIBS, you may be prescribed antidiarrheal medicine such as loperamide or Imodium. Additionally, 3 cyclic antidepressants such as amitriptyline may be prescribed in small doses to treat IBS symptoms. This type of trip may be effective in diarrhea predominant IBS, and it helps to slow bowel movements. A non-prescription medication option is probiotics, which help to boost friendly bacteria in digestive tracts. There is some evidence that probiotic helps to reduce symptoms of IBS, or the other is dietary changes and psychological treatments. Thank you.
Very nice. That's a very complete list of therapeutic options. I think the other thing that is really important, I mean, I'm not a clinician, but I interact a lot with colleagues who take care of patients like this, and reassuring the patient that there isn't anything majorly wrong, and that if this is indeed post-infectious IBS, that the natural history of the disease that we know from these large outbreaks means that she will probably eventually recover and regain her normal bowel habits. But symptomatic treatment, as you suggested, is really the mainstay of taking care of these patients. We don't have a lot, particularly for diarrhea predominant IBS, to treat right now.
Thank you, Professor.
Okay, thank you, Prof. Kim, for the explanation. Such good knowledge for us. And again, thanks for your time.
And thank you for introducing your excellent students.
Thank you, Prof. Dr. Widya, terima kasih.
Thank you, Dr. Dodi Taruna. And also thank you, Calvin, Anal, and also Rahaf, for providing a very extensive answer about the pathophysiology of diarrhea and its relation with the gastrointestinal physiology function. Thank you, Prof. Barrett. And now the last set will be given to the student from University of Jember. I invite Dr. Adelia Handoko to lead this session. Your time is 8 minutes, Doctor.
Okay, thank you. Good evening, Prof. Good morning, student. Okay, ready? Okay, there are 3 questions, and the first question, before answer, you can read maybe 30 seconds. Okay, the first question. Okay, first, Ali, please. Can you hear me, Ali?
Yes, doctor.
Okay. Please answer.
Thank you for the opportunity that has been given to me. Based on what Prof. Barrett mentioned in the PowerPoint, dietary lipid is divided by triacylglycerol and cholesterol ester. Actually, it's divided by 3. One more is phospholipid. And after digested and emulsified, triacylglycerol breaks to monoacylglycerol and fatty acid, and cholesterol ester breaks to cholesterol and fatty acid. And for the uptake of monoacylglycerol and fatty acid can be facilitated by protein such as CD36 and fatty acid transport protein 5 or FATP5. And for the cholesterol uptake is mediated by Niemann-Pick C1-like 1 protein or NPC1L1. And there are cholesterol efflux mediated by ABCG5 and ABCG8 to limit net intestinal cholesterol absorption. Thank you. I think that's all from me.
Okay, thank you, Ali. Prof. Barrett, maybe review the Ali answer.
Absolutely perfect, Ali. Very nice summary. And I think it's really interesting how we now know that these are protein-mediated effects. We always suspected this because the transport was saturable and appeared to be a sort of protein-mediated process, but we now have a lot more information about the precise molecules, and that gives us extra information about how to intervene. So thank you, Ali, for your answer.
Okay, thank you, Ali. Ali background is our campus in Jember, Prof. Barrett.
Nice.
Okay, the next question. Okay, Sabita, please open your mic.
Okay. Thank you for the opportunity. Cholesterol is contained in bile salts, plays a role in the digestion of fat-soluble vitamins. In addition, cholesterol is an intermediate material for the formation of a number of important components in the body, such as adrenaline, the hormone estrogen, testosterone, and bile acids. In addition, cholesterol is useful for helping the body break down fat in the digestive tract so that it can be absorbed by the body. Control of cholesterol homeostasis by enterohepatic bile transport is the role of feedback mechanisms. So cholesterol homeostasis is achieved through tight regulation between synthesis, dietary absorption, utilization of bile salts, and excretion. These processes are regulated through 3 non-feedback mechanisms, namely autonegative regulation of hepatic bile salt synthesis and positive regulation of intestinal bile salts and cholesterol absorption and excretion. Then what happens when there is excess or lack of cholesterol in the body? Excess cholesterol can lead to heart disease, stroke, and cancer, and etc. Whereas a lack of cholesterol can result in abnormal homeostasis in the body, a model for enterohepatic cholesterol metabolism in conjunction with dietary inputs, where for cholesterol was used to obtain insights into the role of the feedback. The analysis demonstrated the significance of the negative feedback in maintaining psychological levels of cholesterol. Furthermore, the positive feedback by the intestinal bile salts on the cholesterol absorption or excretion processes plays an important role in plasma cholesterol homeostasis. Under familial hypercholemia, perturbations in the hepatointestinal reversible bile transport revealed that an increase in the transport of bile salts from the intestine to the liver decreased the hepatic cholesterol absorption rate. Under such a condition, a twofold decrease in intestinal bile salts transport resulted in approximately 20% reduction in the plasma cholesterol level, thereby restoring it to normalcy. This suggests that bile transport control strategy presents an alternative therapeutic method that can effectively reduce cholesterol absorption to attain cholesterol homeostasis. I think that's all for me. Thank you.
Okay, thank you, Sabita. How about the answer, Prof. Barrett?
I think that was a really fantastic answer. And this was not a very fair question because there is so much to say. There's people who've gotten the Nobel Prize to figure this out. So you did a wonderful job and thank you. You should write a short article about this and try and get it published because you did a really nice job. Thank you.
Okay, thank you, Sabita. Okay, for the last one, the third question, please, Natasha.
Okay, thank you, doctor and professors and all the panelists for the opportunity. So I'll answer the question directly. So intestinal lipid transporters are potential pharmacological target to reduce the lipid absorption. As mentioned before by Professor Barrett, one of the transporters is Niemann-Pick C1-like 1 or NPC1L1. NPC1L1 is expressed on the enterocyte or gut lumen as well as the hepatobiliary interface and plays a role in facilitating internalization of free cholesterol into the enterocyte. And this transporter was identified as the primary target of ezetimibe. Ezetimibe selectively blocks the NPC1L1 protein in the jejunal breast border, reducing the uptake of intestinal lumen, micelles into the enterocyte, and increase the clearance of cholesterol from the blood. And the other potential pharmacological target is ABCG5 or ABCG8. As mentioned before, that the upregulation of ABCG5 and ABCG8 by metformin could also be of benefit in disease of cholesterol excess. That's all my answer. Thank you.
Very nice. Thank you. You obviously learned from the lecture. That was very impressive.
Okay, thank you, Prof. Barrett. Okay, thank you for Ali, Sabita, and Natasha, and thank you, Prof. Barrett, from the lecture. Okay, I will give back to Dr. Widya. Thank you, Dr. Widya.
Thank you, Dr. Adelia Handoko, for leading this session, and congratulations for the fantastic answer for your smart student, Ali, Sabita, and Natasha. Thank you, Prof. Barrett, also for your feedback. Ladies and gentlemen, now we have 5 to 10 minutes for question from the audience. I would like to invite the audience to raise your hand if you have any question for Prof. Barrett. Maybe Aulia, can you help me? Is there any participants or audience who raised their hand?
There is no question until now. So maybe for the attendance, especially for students, if you have any question, please ask Prof. Kim Barrett in this great opportunity. There is one, Dr. Widya, Muhammad Rajawali. I will allow Muhammad Rajawali as panelist.
Please.
Yeah. Already in panelist session, Dr. Widya.
Yeah. Hello, Prof. It's a great opportunity to watch your lecture. It's interesting. What I want to ask is about the bile, the bilirubin. When the bile become conjugated by the microbes in the gut, I forget it become conjugated or unconjugated. But how could the bilirubin, the unconjugated bilirubin, absorb back to the circulation?
Yeah.
Yeah. I want to ask about that because yeah.
That's a very good question. So the bilirubin is conjugated in the hepatocyte, and part of the reason that it is conjugated is to allow it to be excreted from the body, because the conjugated bilirubin is not readily reabsorbed across the colonic epithelium. But the bacteria in the colon can deconjugate the bilirubin so that you have free bilirubin again, and then that can be passively transported across the epithelium. Does this make sense?
So after conjugated, it become unconjugated again to brought back to the circulation.
Yes. Yeah.
May I know from which channel or which door it could be brought back to the circulation? Is there any?
There are specific transporters for the conjugated bilirubin to export it from the liver. I believe that the unconjugated bilirubin can be passively reabsorbed, so it doesn't need a transporter specifically.
Okay. Thank you, Prof.
You're welcome.
Thank you, Prof. Kim, and also thank you, Rajawali, for your question. Maybe we can invite one more question.
Yes,
for audience.
there is Tatiana from University of Pajajaran.
Please.
Can you open your video and unmute, Miss Tatiana?
Yes, am I audible right now?
Okay.
First foremost, thank you for your insightful presentation, Prof. Kim Barrett. And there's one interesting topic that I would like to ask about the fecal transport. So are there any requirements for fecal transport other than healthy family donors, especially like there's a possibility that the patient is like over age and they don't have any relatives, and how to solve that situation? Thank you, Professor.
That's a very good question. And I think that there is a lot of people studying this right now. The donor needs to be quite carefully screened to make sure that they don't have some sort of pathogenic infection. But there is a lot of centers that are finding donors that are particularly effective with the fecal transplant, and then they are banking their stool, so they have a lot. But there are also centers that are trying to make a synthetic stool. So they actually take the bacteria and they don't have to use the fecal material. They are trying to make collections of bacteria and using that instead. So it's an area of extremely active clinical investigation right now to really understand exactly what is of benefit and how to best transfer it. But very good question.
Thank you, Professor Kim, for your answer.
You're welcome.
Thank you, Prof. Kim. Thank you, Tatiana, for your question. Is there any question or you limited time, Mbak Aulia?
The time is over, Dr. Widya, and we don't have any question anymore.
Thank you, Mbak Aulia. Thank you, Prof. Kim Barrett, for stimulating our curiosity and widening our insight due to a limited time. We do apologize that we cannot answer all of the questions. Distinguished professor, college, and also student, now we have come to the end of this session. Prof. Barrett taught us current and comprehensive knowledge in gastrointestinal physiology, how molecular and also cellular interact and influence the function of digestive tract, and also its application on health problem. I believe that this event has enlightened all of us and inspired us to never stop learning. Once again, thank you very much for your time, Prof. Kim Barrett. And for students, we will announce the prize winner by email soon. On behalf of Indonesian Physiology Society, I would like to say thank you for everyone who participates and support this event. Of course, Prof. Kim Barrett as our special guest lecturer today. and then Professor Dr. Ketut Tirtayasa as the leader, and also all team members of Pendidikan Physiology of IPS, Elsevier Publisher for providing the book of crash course System Gastrointestinal for the prize, and also judges of the student awards. We have Prof. Ketut Tirtayasa from University of Udayana, Dr. Minarma Siagian from University of Indonesia, Dr. Denny Agustiningsih from Gajah Mada University, Dr. Hardian from University of Diponegoro, and Dr. Irfanuddin from University of Sriwijaya. And also, of course, IPS Chief of North Sumatra, Bandung, Surabaya, and Jember branches, all lecturers who lead question and answer session, all IPS members, and also students and other participants. We are in a great hope that we could maintain the spirit of sharing and caring among others, member of IPS. We'll see you again in other occasional. Thank you and have a good day. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Picture, please.
Bro.
Thank you, bro.
Thank you,
Can
Professor
we have a
Barrett.
picture?
Thank you so much,
Prof?
Prof.